Good morning, everyone. I am Wataru Kamitani from Department of Infectious Diseases and Host Defense, Graduate School of Medicine, Gunma University. Thank you for having me at Fukuoka One Health Forum. In this forum, as a part of workshop on zoonosis, I'm going to talk about SARS or MERS and the COVID-19 and coronavirus that is often a major topic recently. Now moving on to the next slide. To start with, I'd like to talk about One Health. As you may already know, its direct translation means single health. What this means is that in order to protect human health, we also need to pay attention to animals and environment. It's not just human health, but also to protect animal health, we need to pay attention to its environment and the health of the humans that surround the animal. That is the concept. And here is the schematic. We have humans and animals, and then we have surrounding environment around humans and animals, and all of this should be considered as one. We have to pay attention to all of these and think about the health of humans, animals, as well as environment. That is the concept of One Health. You might wonder, why is it that coronavirus, the topic today, falls into the category of One Health? And that is because coronavirus, in fact, relates to zoonosis. Earlier, I talked about the concept of One Health. So you have humans, animals, and environment. In case of infections, when infectious diseases go back and forth between humans and animals, then such diseases should be considered from the point of view of One Health for the infectious control. Moving on to the next slide. In this slide, it briefly explains the outline of coronavirus. It belongs to order Nidovirales, family Coronaviridae, and there are alpha, beta, gamma, and delta genera. It is a positive single-stranded RNA. It is a very long 30,000 bases of RNA. How long, you might wonder? There are many RNA viruses, and there are many viruses with RNA genes, and among them, the longest genetic information is what the coronavirus has, and it's one of the features. And you have often seen these photos of coronavirus on mass media. You have spikes protruding out, and this looks like solar corona, and that's why it is named coronavirus. As I said before, there are alpha, beta, gamma, and delta, 4 genera. Gamma, delta are in the area of poultry, includes very important viruses, but gamma and delta in principle infect among birds. On the other hand, if you look at alpha coronavirus, we have human coronavirus 22QE, which causes human flu. And in the alpha coronavirus, there are viruses that infect cats, dogs, and swines. In beta coronavirus, we have mouse hepatitis virus. This virus has been studied for long, and in studying coronavirus, mouse hepatitis virus study has provided us with very important knowledge on coronavirus. In humans, we have 3 types of coronaviruses which have been very problematic. In 2002, we had severe acute respiratory syndrome called SARS. The SARS coronavirus started in 2002. It started in China and spread across the world. It lasted for a year. And then we have MERS, or Middle East Respiratory Syndrome virus. It started in Middle East in 2012. And then we have COVID-19. COVID-19 is the name of the disease, and the virus for COVID-19 is called SARS-CoV-2. So as you can see, SARS coronavirus in 2002 and the current virus are very close. As I said, when you think about coronavirus, it's important to think about One Health. For example, in case of SARS coronavirus in 2003, it originally was carried by bats, and then it entered into the human world, but in between we had masked palm civet, so it was carried to humans from masked palm civet. In case of MERS, dromedary was the natural host, so the infection came from dromedary to humans. From this, we can say that coronavirus not only infects humans. In case of viruses that cause serious pneumonia among humans, they came from bats and other animals and then to humans. So to control these diseases, the concept of One Health is very, very important. Now let's look at some of the diseases caused by coronavirus. By viruses from coronavirus, respiratory diseases and gastrointestinal diseases are developed. The current COVID-19 infects the respiratory system, causing pneumonia, but there are other coronaviruses infecting animals. They impact not only respiratory system, but they also have cause diarrhea among swine. That is the type of virus that would have damages and gastrointestinal diseases. So there are 2 types of coronavirus. One causes damage on respiratory system, the other causes damage on gastrointestinal system. The current virus, in terms of COVID-19, it mostly affects respiratory system, but we also found virus in gastrointestinal system. So coronavirus easily infects respiratory system and gastrointestinal system, and the COVID-19 mostly affects respiratory system. As far as coronavirus is concerned, now it is a major topic across the world, but in the past, researchers did not think that coronavirus was this much of a major importance. When I was a student, I wanted to study coronavirus abroad, and my mentor at graduate school, when I told him I wanted to study coronavirus overseas, well, at that time, coronavirus was a major topic as there was an outbreak in 2003, but it ended in a single year. So my professor said coronavirus ended in 2003. And I was just shocked to hear that. And the professor said that even upon your return from overseas, it's difficult to find a position here in Japan if you are studying coronavirus overseas. That's how little people paid attention to coronavirus in the past. Now, if you look at the history of the discovery of coronaviruses, As you can see, it is summarized in this slide. In 1931, infectious bronchitis virus that infects poultry was first discovered. Then, in 1940, mouse hepatitis virus and transmissible gastroenteritis of swine virus were discovered, and in 1960, human coronavirus 22QE was discovered, followed by the discovery of OC43, and from a taxonomy point of view, in 1968, coronavirus was mentioned. This is the time when electron microscope was widely used. Then, later, in 2003, we had SARS coronavirus 1, and in 2012, we had MERS coronavirus, and in 2019, we had SARS coronavirus 2 to be discovered. In between SARS-CoV-1 and MERS virus, we had new viruses like human coronavirus NL63 and HKU1, but they were not as impactful as SARS and MERS. So as you can see, as far as coronavirus is concerned, first there were coronavirus impacting animals, but gradually we started to see human coronaviruses, and starting from year 2000, once in a decade we see a coronavirus discovered, and currently we are in the state of pandemic. This is about SARS-CoV-2, which is currently very prevalent. If you look at cases, this is data from the website as of the 4th of November 2020. Already close to 48 million people were infected globally. Among them, 1.2 million people have died. If you look at the world map, red shows where the patients were identified. As you can see, we have North America, South America, Europe, India, and Asia. So in many parts of the world, we have infections identified by this virus. So once incorporated in the cell, coronavirus try to express its own genetic information. In this case, it is the positive RNA, so you have the non-structural proteins. 16 of NSP are made. These NSPs have various functions, including genomic replication or messenger RNA synthesis. And using the RNA genetic information in Golgi and in endoplasmic reticulum, particles are made with bedding in ERGIC. They are released outside. Once released, the virus finds a new cell to infect. This is the genetic structure of coronavirus. Red and brown are non-structural proteins that play important role in genome replication of the virus. Shown in blue are proteins that make particles. We have proteins S, E, and M, and these three make particles in which genetic information is incorporated. The spike protein is important for the binding, so we should develop a drug or vaccine for that area to stop the infection. Or if you can target the NSP, which play an important role in genomic replication, such a drug can stop the replication of the virus and could be a very effective medicine. Now, once again, let's talk about the history of coronavirus. About 20 years ago, in 2003, we had a SARS. In those days, people did not expect the coronavirus to cause this severe respiratory disease, so it was a shock to everyone. It's called severe acute respiratory syndrome, or SARS. Since this happened about 20 years ago, people, I think, are not very much remembering what happened then. But actually, if you look at the occurrence of SARS, orange shows where the infections were reported. Just like the current COVID-19, it spread all over the world. But luckily, this virus disappeared from the world in just a single year. Now, if you look at the symptoms of SARS, you have acute fever of over 38 degrees centigrade. Then you have cough and dyspnea, symptoms of pneumonia. The pathogen is SARS coronavirus. In those days, there was either one or two not in existence, but now it's called SARS-CoV-1, and it is a droplet transmission type of virus through saliva when you're talking. And then the natural host is, as you can see here, a bat. And we also have masked palm civet that intermediates the transmission to humans. In case of SARS, 80-90% of the people infected are of mild disease, but elderly people or people with underlying diseases may be more severe, and the mortality is around 10%. And about 10 years ago, in 2012, MERS started in Middle East. So it was in the Middle East. It's called Middle East Respiratory Syndrome. Sometimes it's misspelled as MARS. Since, as the name tells, it is from Middle East, far from Japan, and most people may not have paid much attention because of the distance. But if you go to international airports, including Narita, you may have seen a brochure warning you about MERS like this one. The symptoms of MERS are like SARS. You have fever, cough, and pneumonia-like symptoms. It's droplet transmission. However, the natural host of MERS is dromedary. The mortality is around 30 to 40%. One important fact about MERS coronavirus is that, in fact, this virus started in camels in Africa or camels in Middle East. Then the virus was infected and humans were infected, and then we have infections in North America and Europe. The MERS started in 2012, and In fact, in 2015, there was a case of MERS. A person who returned to Korea went to a hospital, but this person was not diagnosed as MERS, unfortunately, so he moved from one hospital to the other. In the meantime, the infection spread. This tells us the importance of general public health organizations, medical professionals to be aware of the most recent infection-related information. This is a key for good public health control. And in fact, with this lesson learned, Korea became very careful about MERS. There was another case important, but the same thing was not repeated, and there was a good control of the disease. In fact, when it comes to MERS, it was said that camels were natural hosts of the virus. But you have lots of camels. That was my image of the Middle East. So when I first read the paper, I just wondered if it really was true that the camel was the host. But as you look at this experimental infection of the camel, you see that the nose is running, and histologically the brown shows existence of virus, so there are viruses in the histology. So camel does carry MERS coronavirus, according to this report. In Japan, there are dromedaries, but according to an expert from the Institute of Infectious Disease, no dromedaries in Japan are affected by coronavirus. This shows the cases of MERS reported, and this is from the website of WHO. It started in 2012 to 2013, and we have many cases in 2014, but gradually we see a decline in the confirmed cases. But every year we have some peaks, as you can see. This is 2019, and after 2019 we don't get much information from the WHO website. And earlier I showed you the SARS-CoV-2 infections globally, and you can see that Middle East was also affected by COVID-19. So I think there was more effort going on with the control of COVID-19, and that may be the reason why we don't get the most recent information on MERS. And in 2020, we have COVID-19. This is the name of the disease. It comes from coronavirus disease 19. The causative virus is SARS coronavirus 2. Unfortunately, this is the most recent virus, which is now existing in close proximity to us. It first started in Wuhan, China, and it spread all over the world, and there were strains in Europe. It seems like the virus in Japan seemed to come from the strain in Europe. Various tools are used for rapid genetic analysis of coronavirus, and if you look at SARS-CoV-2, this is the simplified explanation about mutation. This is also from website. This is 2020 in January. First we had yellow and orange. Now gradually yellow disappears, orange disappears, and we have blue, green, and darker blue. So there's a shift of the virus in terms of its mutation. And finally, I'd like to talk about the importance of the basic research for further understanding of the coronavirus. One of the most important tools to study virus is the manipulation system of viral gene. What we do is we artificially make genes, incorporate that in the cell so that we can get a recombinant virus. Of course, we don't mean to prepare noxious virus, but the aim of this manipulation is to see how the virus is replicated, how the disease develops, and we need to identify the functional area of the gene involved in these steps and processes. Of course, this type of technology is very useful for vaccine development and also for the development of antiviral medicine in a very rapid manner. This also is very effective and important for the development of viral vector. What we do in a simplified manner is that we have virus and you take up the gene and artificially you put the gene in the vector. Here we use BAC, and then this is incorporated, introduced in the cell. We have genome RNA and genomic replication takes place for protein to be produced for coronavirus. And this shows how such genetic manipulation is utilized. How to understand the functions. This is just one example. Viral RNA, and we have genome, and then you have protein. This protein, in fact, is called NSP1, and it has a high binding affinity with viral RNA. Why is it that this protein must bind with viral RNA? To understand that, we need the genetic manipulation that I referred to earlier. If this protein cannot bind with the virus RNA, if we can prepare that kind of viral RNA, then virus which cannot be bound will not be able to synthesize many viruses. With the binding, you can make many viruses, but without binding, such virus will have a very poor proliferation. What this means is that if you can inhibit this site where we have the binding of RNA and NSP1, then this could be a good target for drug discovery. And this virus has less replication, so it could be a good candidate for vaccine. That's what we think. Another important thing is that this NSP1 protein is preserved when binding with RNA. So we can find commonality, and by finding commonalities, then we have all coronaviruses and emerging coronaviruses, and we can develop strategies for new medicine. That is our view in our group for our research. With one last minute, I'd like to once again go back to the topic of One Health. Coronavirus is seen from the zoonotic point of view. SARS-CoV-2 started from bat, and MERS have camel as the host, but further back you have bat, and then we have OC43, which is an old coronavirus. In the 1800s, it came into the human world from oxes. And then we have Delta coronavirus, which infected birds, but we have evidence that it has entered into the world of swine. So it came from birds to mammals. So it's possible that this virus could come into the world of humans. So for coronavirus infectious control, the concept of One Health is very important. Finally, I'm doing my research in Goomba University. It's rich in nature. I hope to continue working on coronavirus here, and we are looking forward to collaboration with industry. If you are interested in research, if you are students, please let me know. And this is the end of my talk. Thank you very much.